So I'm interested in Mars biosignatures, and in this study, I'm looking at biosignature preservation potential in an interesting field site not far from where I work at Arizona State University called Verde Valley. And so I'll try to put a little bit of a definition to that very nebulous concept of biosignature preservation potential. If you've looked at some of the NASA documents that talk about priorities for the next decade of Mars science, like the Decadal Survey, like the Astrobiology Roadmap, and like the Mars Exploration Group, the MEPAG document, you'll see that they emphasize sort of the same themes, characterizing aqueous environments, understanding their habitability, looking at biosignature preservation potential. They don't really define it, let alone put constraints on that concept, and using instruments that are going to be on Mars rovers doing this kind of science remotely. And so these are the kind of themes that motivated this study. And if you've looked at the Mars science program in the last decade or so, you'll see that it's been largely characterized by following the water. So, understanding these aqueous habitable environments. And it's funny, some of the press releases, you'll see that come out of this topic, like, near neutral pH found in Gale Crater, things like that. And some of my favorite social media responses to these have been things like, how many times in a week are we going to find water on Mars? Not that it's not very important to understand, habitability in terms of aqueous environments, because I'll talk more about why that's important on the next slide. But I think we should be sort of thinking about the next phase in Mars biosignature detection. So I propose that we should be following the preservation. And the reason why I say that is because my favorite biosignature is fossil biosignature called kerogen. So kerogen is basically what happens to carbon when it gets fossilized and preserved in sediment, terrestrial sediment. If you count the carbon atoms that are, that exist in like living biota today versus the carbon atoms that are preserved in sediment over time, you'll see that there are about 10,000 times more carbon atoms in the sediment. So just statistically alone, it's kind of one good reason why I like the fossil biosignatures. And so this diagram is kind of a generalized schematic of how kerogen, this fossil organics, evolves over time. It's not a very linear process, but thanks to the petroleum industry, who's motivated by organics in sediment for other reasons, we understand a lot about this kind of fossil biosignature. So basically, this is why the Mars community cares about aqueous environments, because they sort of deliver these microorganisms to their final resting spot, so to say, in sediment, kind of concentrate them, like in a delta, sort of a water delta environment or something similar. And if they're lucky enough to be in a certain kind of environment, they get buried and preserved. And over time, with increasing temperature, depth, and burial over geological time, over diagenesis, which is basically, the evolving geological environment in a certain area, you'll get basically this general trend of the carbon atoms. What they want to do is form these 6 carbon rings, and they want to compact and layer themselves like the mineral graphite. And so that's why this process is called graphitization. So the more mature kerogen will actually resemble the mineral graphite, whereas some of the younger stuff will have more of the different functional groups in it. And like I said, kerogen is really, it could be any evolution stage along this process. That's just kind of an idea of what happens to the organics. And environments like evaporitic environments are known to have high biosignature preservation potential. Minerals like gypsum, which is calcium sulfate, halite, which is NaCl salt, are thought to have good biosignature preservation potential. And so it's not surprising that those are the kinds of environments that my field site is attempting to study. My study is attempting to learn more about in the field site. So we are attempting to address some of these ideas about preservation potential through diagenesis in this field site, which is called Verde Valley, Arizona. Like I said, it's a couple hours away from Arizona State University. It's a lacustrine environment, which means it was once a standing body of water, a lake essentially. high in these evaporite minerals. And it's conveniently very similar to where we are right now with the Mars Science Laboratory on Gale Crater, Mars, and that little star there. And the Mars community has also been telling us for quite some time, they've been recommending going to these evaporite-rich lacustrine environments because they seem to be good environments to study for biosignature preservation potential. So what we wanted to do in our study is kind of define this long-term model of biosignatory preservation potential and how it changes over time in one of these environments that has undergone a lot of diagenetic alteration. And see if we can actually create a model of this system. So we've been out to the field a couple times. That's my advisor, Jack Farmer, climbing up a little hill like a kid in a candy store looking at all the exciting evaporitic minerals. We took the samples back to the lab. Of course, every technique that we exposed them to requires different sample preparation, and unfortunately, I can't get into the details of all of that. But the point is, we exposed it to actually our samples to actually five techniques. So petrographic microscopy is basically when you have a thin section of your sample and you look at it under a petrographic microscope, which allows you to see really pretty minerals and colors and textures, and you can use different filters to basically understand how the minerals interact with each other and how the organics are actually preserved within those environments. And so how that system evolves over time, what came first, what mineral became altered to something else, and what the organics are in. And I'll show you another example of that on the next slide, but it's actually a powerful technique. X-ray diffraction, XRD, is a bulk mineralogy technique, which basically allows you to get the main components, mineralogical components of your system. And Raman spectroscopy, now that's an important one because we are actually going to be sending not one but two Raman instruments for the first time to Mars in the upcoming couple years. European Space Agency's ExoMars rover is going to have a Raman instrument, and I believe that's going up in 2018. And then the NASA 2020 Mars rover is going to have a similar type of instrument, a UV Raman, going up for the second time to Mars. So now is a really good time to do these types of analog studies, looking at Mars analog environments and exposing them to Raman spectroscopy and understanding these kinds of questions. And if you want to look at biosignature preservation, a good general technique is also doing total organic carbon content. And basically what you do is you combust your sample and you count the carbon atoms that come off. And you can, look at the different phases in your system and compare, use it as a comparative technique to say, this mineral phase has higher or lower total organic carbon TOC than another one. So that's a good way of kind of gauging relatively. And what that picture is actually under the TOC is an electron microprobe carbon map. So electron microprobe allows you to map two-dimensional surfaces on a thin section and basically get elemental abundances. So that's a carbon map. So the red areas are lighting up in high in carbon. And so I kind of want to couple these techniques, produce integrated data suites looking at preservation potential along the different end-member phases in my system and see if they compare to the TOCs that we're getting. So I'm still kind of working on that. And so even though this is preliminary, we're already seeing a very complex diagenetic sequence in this environment. What we knew about it was that there was a volcano that dammed the Verdi River, which then produced the lacustrine area that the Verdi Basin is in. The first minerals to come out of that system to precipitate were carbonates. So when we put those carbonates, which are these darker areas, when we looked at them under the petrographic microscope, we were seeing these regions here, which are actually gypsum, so calcium sulfate, interacting with these veins of some fluid that went through the system. And then when we looked a little closer at those cavities, we were actually seeing these recrystallized smaller sulfates within that system. So that's actually telling us that the gypsum that was primarily there was dissolved, and then some other sulfates recrystallized in the place of these previous gypsum areas in these cavities. So again, we're seeing alteration, but then when we put these little areas under the Raman system, we were actually seeing interesting excess trace elements, specifically strontium, in our system, and we think they're forming some sort of solid solution with some of the other sulfates. And there are, of course, other diagenetic pathways, but the question is, can we model, kerogen preservation through these different types of sequences? And so another interesting thing about the Raman analysis was that when I looked at that region within the cavity that I just showed you, so when I looked at it under Raman, I saw something like this. The two dark spectra are that same area, and they're the same spectra. I'm just sort of splitting up the Raman plot into two so you can see the different vibrational modes. Raman gives you information about basically the components in your system based on their vibrational modes. So in part of the spectrum, I'm seeing more of a calcium, sodium, strontium sulfate system, because the colored ones are the database matches. So that's matching closer to that, whereas the same sample in other spots of the spectrum matches more of a sodium sulfate system, which is, again, saying something about the paleo environment. And the question is, what? And can Raman be used as a proxy for paleo environments? So that's sort of the next step of this study, to investigate that a little more and to recommend to the community doing studies like this, we should be using more non-end member, non-pure sodium or pure strontium sulfate systems, but solid solution systems, something more in between, to look at realistic data sets in preparation for Raman studies on Mars. And I'm going to leave my summary conclusions up while I take questions. Thanks.
Any questions? Really, no one? So this is really interesting, and I'm curious, so you're talking about the preservation of these sort of through diagenesis, these signatures. Do you have, so you showed some of the preliminary sort of steps as what this sample goes through. Do you have some preliminary results about, I guess you do see kerogen in these samples and you do have preservation?
So we actually did the TOC through the combustion technique. And along this whole system of samples that we've collected, we're actually seeing the most preservation, the highest TOC in the carbonates, so in the surrounding matrix where all of these different sulfates and other evaporates are crystallizing within, which isn't really surprising. I guess the hypothesis there would be as these other evaporitic phases are crystallizing and recrystallizing within the carbonates, they're actually encapsulating less organics, and so the organics are actually, remained in the carbonates. So that's the place to look for the biosignatures.
So where would we find the carbonates on Mars?
Yeah, that's kind of a trick question. That's still under investigation.
Sorry to throw you under the bus on that one.
It's tough to find the carbonates on Mars.
Any other questions? All right, well then let's thank Svetlana again.